Yeah, just to set the agenda, basically, Ohelian has already introduced UniProt, and you already know UniProt in detail, but I'll briefly introduce, just for the premise of the presentation, what is UniProt, the annotation process which we follow, and why we want to use large language models, and our tries with the open source and closed source models, the challenges we are facing, and one of the ways to mitigate the challenge is retrieval augmented generation, RAG system, and then I'll summarize. Briefly, what you see on the screen is the UniProt ecosystem. It consists of several small databases. UniProt knowledge base being our primary database, which consists of the protein sequence and functional information. It consists of two data sets. One is SwissProt, which is manually annotated and reviewed set of protein records, while Tremble being the unreviewed but automatically annotated set of protein records. Then we have UniProt. UniProt is the comprehensive, non-redundant set of sequences. Because sequences might exist in several databases, but we make it non-redundant in UniProt, give it a identifier, and then cross-reference it with the other databases. Proteomes are the set of proteins thought to be expressed by an organism. Usually they are translated by a complete genome and consists of UniProt KB records as well as UniProt. UniRef, it is a clustered set of sequences, so it tries to cover the sequence similarity space at different resolutions, like 100%. So UniRef 100 means 100% sequence similarity, while UniRef 90 means 90% sequence similarity, and so is UniRef 50, 50% sequence similarity. So what is automatic annotation? So there are several computational methods, of which rule-based methods are primarily covering a lot of annotation space. This is just very simplistic view of our pipelines, which are highly distributed and scalable and fully leverage the use of high-performance computing clusters. The left-hand side, the gold, so we use SwissProt. On the left-hand side, you see as the golden-colored database, which is a gold standard database for us because it's manually reviewed. The idea is that we learn the information and express it as knowledge, express it as rules, which are the constructs. So you just imagine rules as something like conditions. So when the conditions are satisfied, the annotation transfer happens. So there are two sort of rule systems we have. One is UniRule, which is the set of rules which are created by human curators. So they learn from SwissProt, make a rule, and finally these rules go through our the annotation pipelines, which take the rules, apply it, and propagate it to the underlying data set, which is the Tremble, which is the unreviewed set. So if you see the sheer scale, it is 244 million Tremble records, which is quite huge. And while the manually reviewed are 571,000. So scalability a lot matters to us. In the another rule system we have is called Arba, which is a fully automatic rule-based system. Where it learns from SwissProt on its own, it creates rules, and then follows the same process as UniRule. The thing I want to draw your attention to is rule confidence. So whenever a rule is formed, it needs to have, for Arba, it needs to have 100% rule confidence, which means that if the 100% of the templates on which the rule is made of, if all of them have the annotation, then only the rule will be applied. Now we have understood the first 2 points, and now I will just go through why we want to use large language models. Go terms, gene ontology terms are one of the functional annotations we use. If you see on the distribution pie chart, there is 60% of the, so these terms, the Go terms are all of the Go terms which exist in SwissProt at the moment. However, despite they exist in SwissProt, Arba has made rules on only 60% of the Go terms. The remaining 39.6% of the Go terms we do not have rules for. And the reason being, for Arba to make a rule on, it needs to have minimum 5 SwissProt templates, plus for the Go annotation, for the Go term, sorry, the rule has to be made on. It needs to have experimental and manual evidences, which is identified by evidence and conclusion ontologies. So basically, even though there are 5 SwissProt templates, there might not be enough experimental evidences. So we want to tap into the 39.6% of the functional annotations which we are missing at the moment. And the way we can do at the moment is to expedite the curation process. So because we want to fetch these experimental and manual evidences, which majorly comes from the literature. So we want to expedite the curation. That's why. So therefore, we define our aims. Our aims to be that we use large language models for manual annotation. How? By summarizing papers for curators with a particular function or gene of interest. Distinguish papers which are bringing in novel information, and then trying to integrate it with the large data sets. So once we have all the information from one paper and several papers, we want to assist the curators as a co-pilot to make accurate, concise, and clear summaries, so that it is easier to curate those Go terms. The second aim is the automatic annotation, where we want to leverage a text mining capability, where we want to extract the functional annotations from the literature and then map it with the already established standardized mappings like Go, subcellular localization, EC numbers. So we tried with the first one, which is Go, which is one of the standardized ontology. This is the prompt we used initially just to try out how it works. So this prompt was run against GPT 3.5, ChatGPT, which we call it. And we asked the GPT to become a UniProt data curator for a centrin protein, which is identified by TGCN1, and focus on the function, subcellular localization, binding, and mutant phenotype. So just to be a curator and focus on these 4 attributes. And this was a summary for this centrin protein from GPT 3.5. It could capture the metal binding properties. Sorry, by the way, with the prompt, we gave a set of sentences from the paper on which the large language model will summarize. So this is a summary, and it could capture the metal binding, subcellular localization, regulatory function, and domains for this protein. Quite exciting. So we wanted to see that how we can apply it at a larger scale for more annotations. Just to understand what is how a curator actually does the Go curation, first the protein of interest is identified. A protein is identified by UniProt accession. The literature is identified. Once the set of papers for which the curator will curate on, the papers are read by curator. And once they see that there are certain Go terms which can be extracted from the literature, they cross-check it with the quick Go database, and then followed by identification of appropriate evidence code, and then it's disseminated to further data sets. We want to plug in, or maybe aid, the third and fourth step in this workflow, where we want to apply large language models to their contextual capabilities to read a paper on behalf of a curator, identify Go terms, and then once we have the Go terms, we can cross-check it with the quick Go database. But we call this process as grounding, which is basically harmonizing the Go terms. So we have the Go terms, we check if they exist, the name and description exists. So now I will go into where our tries with the closed source and open source models. The closed source model being GPT-4, we use the OpenAI API, but the open source models we deployed within our EBI infrastructure to run inference on. So by the way, these models are already available publicly. We're just applying them to extract the annotations. The high-level design of the API looks like this. We have the bottom tier consists of the papers. Then we extract the sentences from the papers, which are talking about that protein or gene. And then we give the prompt as well as the sentences to the large language model. Sorry, this is a very verbose slide, but actually I just wanted to show it goes through a very prompt engineering process. So just focus on the underlying part. So we're asking the prompt to be a data curator, focus on the results section of the paper with the gene, which is parameterized. So we parameterize the gene into this prompt, and then fetch the Go terms which have experimental evidences. The thing to note here is it's a fully end-to-end automatic process. So whatever the result comes from the large language model, it is converted into a JSON structure by the large language model. And the JSON consists of 4 attributes, which is ID name, which is ID means name means the Go term, and ID means the Go term, name means the description of the Go term. The reference, which is the exact sentence from the sentences we have submitted, to prevent the hallucination of facts. So large language models are actually sometimes expert in creating facts. So we are trying to prevent this by capturing the exact sentence. And I'll tell you why later, how we use it. And based on the sentence, which is in the reference, the evidence is generated, which means that why the large language model thinks that the Go term is applicable for this. So we're actually forcing to try the thought process of LLM again. Once these JSON is sort of generated, the reference, we check it in our filtering process that does this sentence exist? If it exists, we let the Go term pass into the next step. In order to have to do evaluation, we created a true data set, which consists of 14 genes. It's a small set. And these are all the recently curated Drosophila entries by a curator. So the last column you see is the PubMed IDs of all the papers, which a curator read for this, but for the gene, which is in the second column. And Go curated are the Go terms which were manually curated by the curator. So the idea is that we will sort of compare these Go terms with the extracted ones. This is the oversimplified view of the pipeline. So we have already discussed the first three steps where we have identified the use case, which is Go term, the paper selection, which the curators gave us the set of papers. The automatic process, the third one is that we extract the sentences from these papers using LangChain. So we focus on the results section of the paper to reduce the prompt size, and then we give it to GPT-4 model. Once we have the terms, there were around 54 terms which were extracted by this model, with around 77% accuracy and recall of 54%. Now the recall is low. The reason we saw with the recall is that it's not the hallucination of facts. Sometimes the large language model has actually hallucinated the description of the Go term. So there is the Go term, but the description does not exist in the Quick Go database. Plus, there were some terms which were obsolete or blacklisted. So because the model has a knowledge cutoff of April 23, and we, for example, ran this in October or September, so there are Go terms which might have been blacklisted or made secondary. In the terms of evaluation, we have, so the true positive consists of if a term, the yellow, so basically the yellow one is the term which is LLM extracted, while the green one is the true term. So if the LLM extracted term is more general, we consider it as true positive, but we have a limit. We do not take to the very highest level of the Go aspect. So there are like 3 aspects in Go. We do not take the super parent terms into this true positivity. If the term exactly matches, then it's a true positive. We have seen cases where the curator has given a Go term from, for example, molecular function aspect, while LLM has extracted a biological process term. They're not necessarily wrong. So if the curator thinks that they're still valid, we consider it as true positive. A false positive is when LLM has gone more specific. We trust the true data set. So if there was a specific term, the curator might have found it. So it be considered as misinformation, therefore false positive. Again, if the term consists in the different Go hierarchy and they do not make sense, it's a false positive. With the GPT-4, the true positive for these 54 terms were 44%. The false positive was 13%. But if you see the hallucination, missed, and obsolete, we were really worried about, like, why is this? Basically, missed terms is because we saw that we only fed the result section. We did not feed the entire paper to the model because of the context limits. Although there are models which have 1 to 88,000 tokens, but there's a rate limit which is applied to these models. So we cannot give the entire paper still at the moment. So therefore, we were focused on result section. There were 4 terms which were marked obsolete. 2 of them were blacklisted. 2 of them were like secondary. So they were removed. Hallucinations, as I talked about, the name and the ID and the name of the Go term did not match. So we have very stringent criteria. We removed them. I'll talk later like how we are trying to mitigate this issue. Then we kept everything same, the first 3 steps, the sentences, selection, paper, everything same, but use Mistral models. Mistral models, 7 billion parameters and 8. These models had very low recall, like 11%. It's abysmal. But basically, they also suffered from the problem of name and description, which were not matching. So basically, we see 3 challenges with this approach. We were facing validity, where the terms do not exist, or their ID and description don't match, or their contextual accuracy is not that great. Basically, the sentences we have fed are not pertinent to the function, processes, cellular component related to that particular gene, or the model has gone wrong. It has merely captured a Go term which was talking about another gene, merely a mention, and it has extracted that term, which is a wrong mapping, I would say. So the evaluation is also a very tricky issue. It's very time consuming, and it takes a lot of time to evaluate these terms. So it is not that scalable. So this is the present design I was talking about. So we have prompt literature, goes to LLM, get the Go terms. The next step we tried was we stored the Go terms. So we wanted to implement RAG system where the Go terms, the ID and the description were vectorized, stored in a vector database. So the sentences which were earlier given directly to the prompt, we run those sentences against this vector database, which, because the vectors are like k nearest neighbors, so it will fetch all those Go terms from the vector database. And then later, we give the prompt, the sentences, and the Go terms which were fetched from the vector store to the LLM. The reason is we want to just use the contextualization capability of large language model, not the named entity relationship, because the named entity relationship is already found by the vector store. So the results were really good. One of the responses is a very small response. I can say it's a function. So the Go terms you see in the red do not exist, but the yellow ones were wrong mapping. But with RAG, although the Go terms reduced, but the validity of the terms was excellent, and they were biologically relevant. So in the end, I would like to summarize that it is a scalable solution to extract the terms, and RAG can optimize it. But we are looking at some sort of benchmarks to how to find which LLM to use. For example, Kappa can maybe have a benchmark which will evaluate these LLM models on the Go functional annotation part. Finally, I want to thank the UniProt team across the 3 sites. It's a hard work from all of them, the funders, and thank you, Anupur, for questions.
Thank you. Questions?
Hi, thank you for the great talk. I have a question about how you handle the randomness in the language models. For example, in GPT, due to the stochastic nature of the GPT, you get different responses for the same question. If you had any training, any specific settings for the temperature or the top P?
Yeah, so you're right. So basically, we use the temperature is really low, 0.2. And the top P was set as 0.8. So we always look at the 80% probability of the tokens. And for each of these each of the tries, I make 2 tries and then average out the Go terms, so which were common in both the tries, and then take those Go terms. So there is relative sort of randomness. But the temperature, if the temperature is low and the top P parameter is set, it can be managed, I would say.
Okay, great.
Yeah.
Thank you.
Yeah.